So welcome to the Western Regional Webinar, our monthly webinars. We are uh, excited to do bringing nanomedicine from the lab to the clinic. Our speaker today is Dr. Ian Villamagna from the Bay Area. As a reminder, we're your North American Western team. I'd like to introduce, as a reminder, we have Angel Galarza, who's our Regional Sales Director. My name is Kim Killian. I'm the Regional Sales Manager in the South, located in San Diego. Viet Nguyen is our Field Application Scientist for the South as well, located in Irvine. And then our speaker today and Angel's Field Application Scientist is Ian Villamagna, and he is located in the Bay Area. We're really excited to launch a new addition to our offerings. We're going to be doing office hours. It'll be routine office hours every month, and you'll receive a notice either for our next webinar or journal club invite. If you scroll to the bottom of the invitation, you'll see a link where you can sign up for office hours. And the team will be meeting once a week at noon Pacific time. And we will open up 60 minutes to your questions. So all you'll have to do is reserve a time slot. And this can be questions you have from our webinars, our journal clubs, or something that's going on with your research. So we're excited to announce that.
Thanks, Kim. I appreciate that. And thank you, everyone, for joining us today. So a lot of return people here coming back for our webinars. We do run these once a month with webinars and then once a month with journal clubs. We really appreciate everyone showing up and bringing their insight and their questions and discussions to these events. I think it makes them much better. We do appreciate you guys joining us. So with that, I will go ahead and bring up what we will be speaking about today. And so today we're going to be talking specifically about scaling up. And this is one of the topics that we get asked a lot about from a lot of our clients is, what does scale up look like and what do I have to do to get to that stage? And it's a really large stage in the development process for nanomedicines. And so it can be difficult to answer these questions with blanket answers. But today what we're going to do is try to kind of put some perspective to the scale-up process and look at some of the key components that go into getting to that scale-up process, and really what we believe the key is to getting there. So, as always, if you've seen any of our presentations, we always like to start with our vision. Our vision at Precision Nanosystems is to accelerate the creation of transformative medicine that significantly impacts human well-being. And we believe scale-up is a big part of this. So getting customers to scale up, getting them to a place where they feel comfortable scaling up, and doing it in a rapid fashion is really what we believe is going to accelerate that process and getting these medicines to the clinic and out to patients that need them. So I think, ever since earlier this year, when we've become really invested in COVID-19 and we've seen it unfold before our eyes, one of the things that came along with it was this major breakthrough, and that's RNA vaccines. And for a lot of us in the field, we know RNA vaccines and nanomedicines and nucleic acid medicines inside of liposomal or lipid nanoparticle carriers are not new. But they are new in the sense that they're now rapidly moving through these phases of FDA trials and into the clinic very quickly. And right now, if we look at what's being done with these RNA vaccines, we have a number of them in various stages of trials, including two of them, so with Moderna and BioNTech, already into phase 3 clinical trials. And so this has been a really large development and has really put nanomedicine as a whole in the spotlight and shown that this is a potential way forward, not only for COVID-19 vaccines, but for a large number of other diseases and other implications that we have. Along with that, though, what this has really shown is that there's a need for this really rapid and effective scale up. And so if you think about COVID-19 again, the amount of doses that we're going to need to sufficiently vaccinate even the population just in the United States or Canada is going to be quite large. And with that, what we need is to have a manufacturing platform that can enable that scale up very rapidly. So it can be tested in the lab and move through that process to get to that number of doses very quickly so that we can combat this pandemic right on at the beginning as quickly as possible. This is something where I think, we've seen a lot of this recently. We've seen a lot of this work go into a laboratory stage. And now that we're moving into these phase 2, phase 3 trials for a lot of these vaccines, I think we've seen the question start to shift to the other end, is that can we realistically make these? And is what we're seeing here in the lab going to be repeatable at a large manufacturing scale? And so that's definitely where we're at right now. And it's become increasingly important as this has gone on. If we look at nanomedicine as a whole, I think what stands out mostly is that these are not simple constructs that we're making to try and treat these diseases or to try and treat COVID-19. These are very complex entities that we're dealing with here. So, lipid nanoparticles or liposomes, instead of just dealing with an active pharmaceutical ingredient now, we, in addition to that, have all of these different non-pharmaceutical ingredients to go along with it. Lipids, polymers, different surfactants, things that we have to factor into that nanomedicine development process. And because of this complexity, what it requires to go along with it is a manufacturing process that can enable that complexity and take that complexity and still allow the production of these nanomedicines in a very quick and meaningful way. When we think about scaling up, we have at Precision Nanosystems what I would really say is a very simple philosophy. And that is essentially that in order to effectively and rapidly scale up nanomedicines from the laboratory all the way up to the clinic, what you really have to do is model the GMP process at earlier scales in any way that you can. So anytime you're working in a laboratory and trying to develop these nanomedicines, you obviously want to do this on a smaller scale than you would for a scale-up purpose. For cost saving, for time saving purposes, the smaller scale is always going to be more effective from that standpoint. But if we can model that GMP system at an earlier stage in the development process, what that enables us to do is take really a large amount of data on that process at a very early stage that allows us to learn from that and build towards a scale-up system. It also allows us to de-risk the entire process. And so by the time we get to the point where we're saying we're ready to go and we're ready to scale up and make large dope batches of this nanomedicine, we've seen most of what we think we have to see. So we've seen all of the issues that could come up with manufacturing. We've really run this nanomedicine through the development process at an earlier stage, enabling us to get to that drug product much quicker. I'd like to kind of talk a little bit about Doxyl because I think it serves as a really good case study. And for anyone that's come to these talks before or has seen me talk, or Viet probably as well, you've probably heard us talk about Doxyl before. And the reason Doxyl gets brought up so much is that it really is a microcosm for everything that's good in the nanomedicine development space, but also a lot of things that can go wrong in that space. So just a little bit about Doxyl. What this is a liposomal formulation of doxorubicin, which is a very strong anti-cancer agent. Doxorubicin was very effective in treating cancer for many years, but it also had a lot of systemic side effects, especially cardiotoxicity, that was limiting its ability to be used. In the mid-90s, what was found, essentially, is that they could package doxorubicin inside of a liposomal carrier like you see here on the screen. And what that allowed for was essentially the delivery of this Doxyl being the liposomal formulation in a systemic fashion. And you still had the effects of doxorubicin, but without all the harmful systemic side effects. So the side effects went far down, and you were able to deliver it to more patients, and more patients were able to use this for a more prolonged period of time to treat their cancers. Doxyl and doxorubicin both worked extremely well, and it was extremely promising. Where Doxyl was plagued by was manufacturing issues. And so all the way up through the mid-2010s, really, Doxyl was plagued by these manufacturing issues, where there were consistent shortages of this medication because the scale-up and because that manufacturing process was not working effectively. If we look at the reasons as to why, with Doxyl specifically, we don't really know right now where that issue came from, or at least I don't. But I think what we can do is look at how nanomedicines are developed, and specifically liposomes, and we can start to get some ideas. And so if we think about traditional liposomal development processes, they usually look something like this, where you have multi-steps here in this process. So what you're looking at here would be a very traditional approach to making liposomes. This is done through some thin film hydration, like you're seeing in the top row, where you're going to actually make these multilamellar vesicles. And in order to have an effective liposome, what you then need to do is do some downsizing, either through sonication or extrusion, microfluidization or homogenization, to get these unilamellar vesicles like you see on the bottom row, which is what your final drug product needs to be. The issue here is that there are a number of steps in this process where something can go wrong. And I think with Doxyl production, what we're probably looking at with the shortages is that it can be very difficult to take this process that you do in a laboratory and scale that to a manufacturing stage. If you compare that with something like the NanoSembler platform, which uses microfluidics, what that allows you to do is essentially take these steps and remove quite a few of them. So for instance, with the nanosembler, as compared to the conventional approach here, you would still have your lipids and your drug combined into a system. They'd run through a microfluidic pathway. And instead of making these multilamellar vesicles from the start, you make these unilamellar vesicles. So you've automatically taken out between 2 to 8 steps in this process, depending on how many times you have to go through that downsizing procedure. Having this reduction in complexity can really reduce the amount of time and the amount of investment that is going into this nanomedicine product. And that allows us to do more development process in the early stages that allows us to model this manufacturing procedure in the long term. As kind of an illustration as to how that would work and how that microfluidic process works for something like Doxyl, you can see here that the essential idea would be that you'd have a PBS buffer in one inlet of the microfluidic channel. It gets met with your lipid mix in ethanol, along with any other components in that liposome, and you're able to form these liposomes that you see on the right. This process happens extremely quickly. The mixing itself happens on the scale of milliseconds, and the entire production of a laboratory scale liposomal formulation can happen in less than a minute. After that fact, you're able to go back and remote load your drug into these liposomes if you're not loading it when you're actually making it. So again, I think what it comes back to really at the end of the day is, how do we model that GMP process at an earlier stage? So how do we look at the manufacturing process for something like these liposomes and do as much as we can on an early stage, so we know when we get to that stage of scaling up and trying to make that drug product, or even sterile filtration, when we're at that final stage, if you will, that we can trust the data we've seen and we can trust our process to carry us forward into the clinic. What I'd like to do now is focus on a publication that I think is really quite good. And it talks about this rapid scale up of active loaded pegylated liposomes. So this is one example of a nanomedicine development process where they walked through this scale up stage, and they walked through quite quickly. The paper comes to us from the Yvonne Perry group at the University of Strathclyde. And essentially what they looked at here were creating liposomes through a microfluidic platform, and really looking at a number of different parameters that they would test in order to get an idea of this liposomal production. Afterwards, they loaded doxorubicin, so much like Doxyl, through an active loading mechanism. And then at the final product, they were able to compare essentially a bench scale, so they're using the standard herringbone mixture, to a larger scale production, and to see how quickly and rapidly they were able to scale to that point. From the very beginning, I think what this paper does really well is it shows us that scale up is all about modeling at a smaller scale. And what the smaller scale really enables us to do as scientists is look at a number of different aspects in this liposomal development process and control for all of them. So they all become part of that actual scale-up process. So here, what we're looking at are a collection of studies that were done on these liposomal formulations at a number of different variables. And so there's a lot of data here in this figure. But essentially, if you look at the top row and the bottom row, they're comparing two different liposomal formulations. And they've done this at different flow rate ratios, different total flow rates, and different lipid concentrations. I think what's important to remember here from data like this is that they're not necessarily looking for one specific formulation, one specific flow rate ratio that they think works well, or one specific total flow rate that they think works well. And that's typically not how a lot of this nanomedicine production and development is done. What I think they're really looking for here is establishing these trends where they can say very readily that as we increase the aqueous to organic flow rate ratio, as you see in that column A on both liposomal formulations, we know that the size of these particles decreases. They do this as well with lipid concentration. So at what stage do we increase the lipid concentration to where we have these higher sizes of these liposomes? When we have all this data and we can take that data with us, what that allows us to do down the line with a scale up is look at these faster flow rates. Does what we're seeing mimic what we saw at a laboratory scale? And this sort of idea enables that rapid scale up. It's important to know that they didn't just look at things like the formulation in terms of concentrations or what can be done with the microfluidic platform, but also other items as well. So looking at things like the different solvents that could be used. So here they compared, for instance, methanol and ethanol, looking at the change in Z average diameter and polydispersity, and looking at the difference there in terms also of different flow rates. Again, it's about using that smaller scale to build as much data as you possibly can on your constructs as early as possible. If you walk into a scale-up process with an arsenal of data behind you, it will make the scale-up process easier insofar as you have enough data to understand what you're seeing at a larger scale, that you don't have to repeat a lot of these studies. You know what you're looking for going into it. They even went as far here to look at temperature. And temperature, I think, is a really interesting thing to look at as well, which is something that I don't think we see as typically. Some lipid mixes are very sensitive to temperatures and need higher temperatures to be formed. Here, what they saw is that depending on the flow rate ratio, they didn't need a specific temperature. And that becomes important with larger batches, with large scale-ups, where it becomes a little bit more difficult to keep your lipid mixes at a certain temperature. Being able to say that we can do something at 20 degrees Celsius rather than 60 if we use this flow rate and still have a liposomal formulation that we're comfortable with at the end can be very effective and can really enable that rapid scale up as well. Again, here they looked at Doxorubicin Release. And so I think this is a key component of this here. If we're making liposomes at a small scale, but they don't actually release that drug and enable that drug to work, then there's really no point in moving forward with looking at the constructs in a scale-up method. And so I think a lot of times these studies are at a weird in-between stage where you've done your development process, you've looked at your particle characteristics or your liposomal characteristics. But studies like these, where you're looking at how efficiently and effectively these liposomal formulations work, are also extremely important and give us an indication about which we should move forward with into scale-up. Here they looked again at different lipid compositions and the effect of that on doxorubicin release. They looked at, again, different flow rate ratios and those different temperatures, and took all that data to look at if there was a difference between any of these and what effect it played on an actual efficacy measure of this liposome, so in terms of doxorubicin release. I think what also is really important, and it really says a lot about the scale-up process in general, is that if you look along the bottom here, for all their studies, they continually go back and do what we call TPQA, or total particle quality analysis. And that consists of size, PDI, and encapsulation efficiency across the board. I say this a lot, and so if you've heard me speak before, you've probably heard me say this, but anytime you do really anything with a nanomedicine development, insofar as changing any variable within that process, it's extremely important to go back and run these tests to look at the change in size and PDI and the encapsulation efficiency. That is, in my opinion, going to be your best measure as to if your process is working or if changing something negatively altered the effect of that particle. If you go through these sorts of experiments looking towards scale up without doing this, what you really have is no way of knowing if what you're doing is negatively affecting these liposomal formulations, which will negatively affect your ability to scale up in the long run. Finally, again, I think it's important to remember that with a scale-up procedure, what you always have to go along with that is downstream processing, especially for these liposomal formulations and for lipid nanoparticle formulation. When you make these formulations with a microfluidic platform, what you typically get at the end is a solution that is roughly 25% ethanol, give or take, depending on the ratios you're using. And what this typically requires before you can go into any sort of animal human study is the removal of that solvent. And so here what they've done is use dialysis as a model for TFF. So TFF, or tangential flow filtration, is something that you're going to use typically on a much larger scale when you have those large batches to effectively and really quickly remove that solvent and do a buffer exchange with it. This will typically increase the stability of your formulations. And so it's important to try and understand from a very early stage, again, how your constructs, your liposomes or your lipid nanoparticles are going to respond to this process. Doing this through dialysis gives us an ability to actually model what it may look like in the TFF procedure. And doing that, again, adds another piece to that data where we can say, we not only know everything there is to know about the constructs of these formulations from all the data we covered in the last couple slides, but we also have modeled that downstream process. So we know what we're looking for when we go into this large scale. We know what's effective on a small scale. How can we apply that to a larger scale? At the end of the day, I think what this paper really showed is that they were able to take their formulations that they did at 12 milliliters per minute and scale them up very rapidly, all the way to 90 milliliters per minute. And at 90 milliliters per minute, what you can realistically see is that you're able to make these formulations at a much quicker rate. And so that enables you to make these large doses that you're going to need for those large-scale animal studies or human clinical trials as you get to that stage. It's important to look, again, at things like the total particle quality analysis. So the size, the PDI, and then the encapsulation efficiency as well, and looking for changes between essentially those when you scale up. And so in between that bench scale, that laboratory scale, and the scale up, do we see an increase in size, PDI, or encapsulation efficiency? One of the things they did here as well, which you see in D, is actually some visual analysis on these particles. So using electron microscopy to look at this particle formation in terms of bench scale and scale up, or lab scale and a scale up model. And that can be another really key characteristic to take and use and look at that to try and understand how this formation is occurring and if this formation is occurring effectively. I'll just close on this specific data set with what this paper described. And so, when I read this paper, what I found really interesting was kind of towards the end, where essentially what they're saying this scale-up process is going to enable is this personalized or just-in-time medicine. So imagine you get sick, you go to the hospital, and very quickly we're able to come up with a brand new formulation that is specific to an individual patient. And the reason why we can say that this is something that's coming our way and something that can be done is that when we can model effectively a lot of these formulations at a smaller scale, when we can take that arsenal of data that we can gather at a laboratory scale, when we enable that rapid scale up, what it allows us to plan for in the future is the ability to make these medications very quickly on that personalized basis as well. And so that's one of the things that they looked at here, and that the group believes is going to be coming in the future.
Ian, before we move on, we had a question in the Q&A about the limitations on the variety of types of particles that can be made on the nanoassembler platform, since you've been talking pretty much about liposomes. The question is Precision working on ways to increase the variability of types of particles that can be made? For example, mixture of polar and non-polar molecules.
Yeah. I think in terms of the platform itself, the platform is an open platform, and so we've seen a number of different types of particles made on this. So we're going to talk about a data set right now, just to close off, about more traditional mRNA-based lipid nanoparticles. But we have a number of customers that are working on polymeric nanoparticles, different types of liposomes with different types of payloads, whether that be small molecules or peptides, lipid nanoparticles that have RNA or DNA. There really are a large number of different types of particles that can be made with this platform. And if there's a specific one, we always ask people to reach out to us, and we have this wealth of publication database that our customers have created that we can point to usually and find something in the realm of what you're looking to do and kind of put you down the right path to lead you towards that development process.
Thank you, Ian. And we have one more question about this specific publication. So if you could go back 4 slides to slide 17.
Yeah, give me 1 second.
Right here, yeah, right here. The question's about the, comparison between these different formulation conditions and the release profiles. Mm-hmm. The low cholesterol group here seems to have the lowest encapsulation efficiency, mm-hmm. And the question here is whether the release data was the percent release data was normalized to the entrapped amount of drug.
That's a really great question. Off the top of my head, I'm not entirely sure. I'd have to go back and check that real quick. So maybe we can get an answer for you towards the end here on that, because I can't remember right now off the top of my head. But I think it is important to note that the lower cholesterol group does have that lower encapsulation efficiency, which is really important as well. So we know cholesterol plays a large role not only on the formation of these particles, and what they can encapsulate, but also how they release. And so looking at the differences in percent release. If I were to harbor a guess right now, I would say that it is normalized, but that's like I mentioned, that's a guess, and perhaps we can get that answer towards the end here, Viet, and try to answer that question.
Thank you, Ian.
Thanks, Viet. All right. So we've talked a lot about liposomes, and I think this plays really well with that last question that we got. One of the other things I'd like to kind of talk about quickly is just a different type of construct that can be made on this platform, and with that scaling up in mind. And so here, what we're going to speak about is the scale up of an mRNA lipid nanoparticle. I think this is especially timely with what we've seen with a lot of these COVID-19 vaccines. So these new vaccines that are RNA-based and have that messenger RNA, and this need for this rapid scale up to get to the amount of doses that we're going to need to vaccinate the population. So this is a model of what a scale up would look like from an mRNA vaccination, if you will. And so what this data looks at here is an EPO encoding mRNA that was packaged inside of a lipid nanoparticle, and was run through a scale-up process in order to look at things much like you saw with that liposomal formulation, but looking at how we can model that process at an earlier stage in a laboratory scale before bringing it up to a scale-up type level. What was done here essentially were 3 different instruments were used to scale up and to make these constructs. So first, the Ignite laboratory scale instrument, in which these constructs were made at 12 milliliters per minute, was used. And as a downstream processing, ultrafiltration was used here for any sort of solvent removal. After that, what was done is using the nanosembler blaze. And the blaze here runs at 60 milliliters per minute and 90 milliliters per minute. So giving us 2 data points to look at in terms of what we see from a scale up at 60 and 90 milliliters per minute. And then finally using ultrafiltration or tangential flow filtration as a downstream process. At the end, what this really led us to was looking at the nanosembler GMP system. And so the GMP system runs up to 200 milliliters per minute. And so it can very quickly make these formulations at a very large scale. You can make essentially upwards of 20 liters of formulation per hour. And we wanted to look at essentially, again, that total particle quality analysis, and was there a change that we saw in any of that analysis as that scale-up process went through. In terms of typical total particle quality analysis, so size, PDI, and encapsulation efficiency, there was no statistical difference whether this was run at 12 milliliters per minute, 60, 90, or 200 milliliters per minute. And I think what this brings back to is that when you use this microfluidic platform, the microfluidic assembly, that microfluidic architecture is retained across the board, whether you're doing this at 12 milliliters per minute, at 90 milliliters per minute, or at 200 milliliters per minute, the same microfluidic architecture is used. And so the same process is being modeled throughout that entire scale up. And that typically will allow us to make these constructs at 12 milliliters per minute and make the same thing at 200 milliliters per minute. Again, for encapsulation efficiency, no statistical difference was noted across the board, regardless of the flow rate used or the system used. But probably most telling here would be the in vivo efficacy data. And so looking at the total serum EPO levels, which would be directly affected by that mRNA that was encapsulated inside of those lipid nanoparticles, across the board, you saw an increased expression and an increased amount of that serum EPO level from every particle population that was delivered, whether it was done at the laboratory scale or at a GMP scale. And then as well with percent hematocrit, which would be directly correlated to that serum EPO level, we saw an increase not only in the different flow rates and the different instruments, but also in the constructs that underwent tangential flow filtration as well. So modeling that downstream process on the blaze, for instance, where you were doing it at 60 milliliters per minute, gave us an indication, and it shows us that that same process occurs at 200 milliliters per minute when you're at that GMP scale. And so with that, just some conclusions here. I think what this really brings us back to is that this platform, the nanosembler platform, and really microfluidics as a whole, what I would say, is that this process is built to de-risk every next step. So doing a formulation at a laboratory scale on the Ignite de-risks that movement into the blaze, which then de-risks that movement into the GMP system. And by doing that in a stepwise fashion and gathering as much data as we can at a laboratory scale or at a preclinical scale with the blaze, what that enables us to do is move into that scale-up phase and move into using that GMP system at a place where we know as much as we can possibly know about these formulations, whether they're liposomes or lipid nanoparticles, and really effectively enable that scale up and that rapid scale up to enable something like getting all those doses out for COVID-19 vaccination.
Ian?
Yes.
We do have a question from the audience about whether there is a way to generalize the scale-up parameter setting, or they need to be kind of optimized case by case, as you're showing the workflow here. Yeah,
it's a great question. I think, my most common answer I give ever is that it's always formulation dependent, because the formulation itself can be very different from one thing to another. And that goes back to the type of cargo that's being encapsulated, the different lipids that are in that mix, the different polymers you're using, whatever it may be. Each formulation is inherently different. And even small changes that we believe are small can have a large effect on the formulation. And so, to a point, I would say there is going to be work that has to be done with every formulation. That being said, I think from a lot of the data that we have from great publications like we saw here from Yvonne Perry's group or some of the serum EPO level data for the mRNA lipid nanoparticle, what you're able to do is really develop a very encompassing experimental plan from the start that enables you to really plan those experiments effectively and move through this very rapidly as well.
I see. And one more, since you're showing the different instrumentation that can be used during the development and scale-up process. Could you tell us a bit more about the differences in chip and channel geometries for these kind of systems?
Yeah, absolutely. So I think it is important to remember here, first and foremost, that across this line that you're seeing on this screen here, the NanoAssembler platform uses what is known as next-gen microfluidics. And so this microfluidic architecture is retained across the platform. It's the same in the Spark as it is in the Ignite, the Blaze, and that GMP system. It does change in terms of geometric scale, which is what next-gen microfluidics allows you to do. It allows you to scale it geometrically, which enables that rapid scale-up. It enables those higher flow rates. But because the architecture remains the same, the physics of mixing of your two fluids is retained across the board. And what that allows us to do is very, very easily model what's going to happen in a GMP system at a laboratory scale on the Ignite.
Thanks, Ian.
Like Kim mentioned off the start, if you're interested in getting in touch with us, our contact information is here. If you're not sure what region you fall in or who to contact, you feel free to contact any one of us and we'll point you towards the correct team to speak to. Please feel free to reach out and get in contact with us if your question wasn't answered or if you think of any more questions that you'd like to talk about. We're always happy to schedule a time to have a call or just through email as well, just answer any questions you might have. I really appreciate everyone's attention. Thank you for joining us. And with that, I'll hand it back to Kim just to close us out.
Great job, Ian. Another fantastic webinar. We want to thank our guests too. August is always a very busy month, hectic with school starting and vacation. So thank you for your continued support. Our next Journal Club will be Friday, September 11th at 9 a.m. Pacific time. We look forward to seeing you then. And just a reminder, we will launch the office hours in September. You'll get an email. And again, as Ian said, you're always free to reach out to us individually. We can schedule a Zoom call or a chat, but these new office hours, they'll just make it easier if you'd like to plan ahead and know that there's a time that we're always available for your questions. So with that, have a great Wednesday. We look forward to seeing you next month, and everyone have a great day. Stay safe and be well.